In this video, we'll show you how to design a plasmid DNA donor template for your homology-directed repair experiment. If you have not already identified a guide RNA and tested it for efficient cutting of your target gene, you should go to the CRISPR design tool before continuing. To design guides for HDR, choose CRISPR site-specific editing to get started. Or if you already know your guide sequence, click on the Order Guide RNA. Alternatively, if you know the DNA sequence around where you would like to make an edit, choose the Design Guide RNA link found here. To design your plasmid DNA donor, start by going to the HDR Donor Design tool. Click on the HDR Donor Designer plasmid option. Start by entering the name of your species or organism in the text box. Then select it from the drop-down menu. Next, enter your gene target by typing the gene name or gene ID, and click to select it from the auto-populated list. For this example, we will choose AKT1. The gene's primary transcript will be displayed by default. Alternate transcripts, if available, can be found in the Transcript drop-down menu. Next, choose the sequence that you want to insert. You can select either MK2, EGFP, or enter a custom sequence. For this example, we will use EGFP. Then, enter or paste your guide RNA targeting sequence. If you have not already designed a guide RNA and tested it for efficient cleavage with the cell type and nuclease being used, you should use the CRISPR design tool to do so before continuing. Finally, select the nuclease that will be used for your gene editing experiment. In addition to the most common Streptococcus pyogenes Cas9, this tool can generate guide RNA sequences for Staph aureus Cas9 and MAD7 nucleases as well. The gene transcript sequence surrounding the guide RNA is displayed. Drag the slider bar, which is green in this case, representing EGFP, to the desired genomic location for insertion of EGFP. Next, click on Generate Primers. 5' and 3' homology arm primer pairs are displayed. Select a forward and reverse for each primer pair. There will only be one option for primers that are adjacent to the insert, since these primers are specific to the exact insert location. On the other hand, the corresponding distal primers have multiple options, listed in rank order, along with the resulting homology arm length. You may select multiple primers to create homology arms of differing lengths. Once you have at least one forward and reverse primer selected for each homology arm, ensure that the Edit-R HDR Plasmid Donor Kit, EGFP, is also selected. It contains the remaining components required to assemble your plasmid donor. If you have previously purchased the kit and are designing primers for a new target, then you may deselect this. Enter a unique name for the primer sets. Lastly, click Add to Cart. You are now prepared to check out with your order. Hope you have enjoyed this video on how to design and order the components to build your DNA donor plasmid for your HDR experiment. More resources can be found in our knowledge base or by clicking on the links below. See you next time.